There are a number of second-generation agents. Ceritinib, again, is an agent that you've been heavily involved in its development. If you could describe that a bit.
Sure. So ceritinib is actually one of 10 next-generation ALK inhibitors that have entered the clinic already. And ceritinib has been developed by Novartis. It actually came from an original tool compound, TAE684, and is a very potent ALK inhibitor, anywhere from 10 to 20 times more potent than crizotinib in inhibiting ALK. Has a little bit of activity against IGF-1R and insulin receptor, although we don't think that at physiologic levels that we're inhibiting those, and really doesn't have any other known targets, other than ROS1 perhaps. But unlike MET, ceritinib does not, unlike crizotinib, ceritinib does not inhibit MET. So this drug has been shown to be quite active in vitro, also against various crizotinib-resistant models, and that really was the basis for taking forward a phase 1 trial. This was a global phase 1 trial of ceritinib in ALK-positive patients. Actually, it was any malignancy with an ALK alteration, but the bulk of patients were ALK-rearranged non-small cell lung cancer. And this trial enrolled, initially we had 130 patients that went into the publication recently, but for the actual filing, it was the full phase 1 study, which was over 250 patients. And overall, what we found is that ceritinib is very, very active in ALK-positive lung cancer, including patients who failed crizotinib already. And it was quite remarkable to see, these patients who have already responded oftentimes beautifully to crizotinib for a year or a couple years, relapsing now, and then actually having a re-response to ceritinib.
And the responses are durable? And
these are durable responses. Median durations of responses are in the 7.5 month range. And the overall response rate for the ALK-positive group, either crizotinib-resistant or crizotinib-naive, is close to 60%. So this data all led to the accelerated approval of ceritinib actually last month.
And there are plans to bring it up front?
That's a good question. The label really focuses or specifies that this is for patients who have failed crizotinib or are intolerant to crizotinib, because the data on crizotinib-naive patients is still very early. So yes, in theory, there could be plans to move ceritinib up front, but we don't have data yet to support doing that. And Ross was just talking about this earlier, about the importance of, what do we need to show in terms of efficacy of ceritinib frontline to really feel comfortable offering that. And you would have to think that probably you need to show that the median PFS with ceritinib as your first ALK TKI is about what we get by doing sequential crizotinib followed by ceritinib.
Again, we're talking about a 6 or 7 month improvement over what we'd see with crizotinib.
That's right. So I would say 16, 18 months with frontline ceritinib.
It's a high hurdle.
It's a high bar.
Very high hurdle. Considering 25 years ago, our median survival, not PFS, was only 6 to 8 months. There are other compounds that are being looked at. Ross, you and I have been involved in the area development. Can you talk a little bit about the AP26113?
So 26113 is another second-generation ALK inhibitor. It may have the broader spectrum of activity against some of the resistance mutations which have been described. It is probably earlier in development. It's a much smaller company and hasn't probably got quite as deep pockets, but we're seeing similar very good response rates, about 60–65% durable responses, clear evidence of activity in the CNS, and that, we should talk about this in some detail. One of the issues is how well do you capture data in terms of activity in the CNS, and everybody's really playing catch up as we start to realize this is a battleground. The ARIAD data currently their data that they've shown is about 8 out of 10 patients have shown responses in the CNS, and they appear to be durable. So
the CNS responding in parallel with extracranial disease.
Yeah, so one of the issues we should touch on that is crizotinib's sort of partial Achilles heel is relatively low penetration of the drug into the central nervous system, and consequently about 50% of patients who progress on crizotinib are actually progressing in the brain. And it's not an adaptation of the biology, it's an undertreated part of the body.
It's my impression that the toxicity profiles of these compounds are different, that with the ARIAD compound, pulmonary toxicity, interstitial lung toxicity has been a greater concern. But the ceritinib compound also has its own spectrum of side effects, particularly GI toxicity. Right. Talk about some of the strategies to reduce that, to mitigate that.
Sure. So yes, ceritinib does cause quite a bit more GI issues than crizotinib, or I would say many of the other next-generation inhibitors like the ARIAD compounds. Diarrhea, nausea, vomiting are each seen in over 80% of patients who were treated at the maximum tolerated dose of 750. So we do a lot of counseling ahead of time. Patients are aware of doing your typical management, antidiarrheals, antiemetics. But to be honest, I feel the best way of managing toxicity when it occurs to that degree really is dose reduction. So I very aggressively dose reduce patients. They go down to 600 milligrams and then even down to 450 milligrams. And I feel very comfortable doing that because in the phase 1 study where we were enrolling starting at 50 milligrams, we were seeing great responses even at 300 milligrams and up.
So 750 may be perhaps too high a dose, or at least
it's quite high for most patients to tolerate.
And the median dose delivered is in the 600 range?
It is. I mean, over 60% of patients are dose reduced, and that actually was sort of early on, and I feel with longer follow-up now, I'm seeing probably close to 80% of patients being dose reduced.